Hello, thanks for stopping by our poster. I'm Dr. Chandrasekhar Honrao from Olaris, and I will be presenting results from our study on identifying metabolic biomarkers of response to CDK4/6 treatment and resistance in MCF7 breast cancer cell line. This work is done in collaboration with Dr. Rina Jesselson and her team at Dana-Farber Cancer Institute here in Boston. So, what is the clinical need here? As we know, CDK4/6 inhibitors have significantly improved outcomes in patients with metastatic hormone receptor positive and HER2 negative breast cancer. Yet, almost 20% of patients never respond to the treatment, and all patients eventually develop acquired resistance. At present, there are no clinically validated biomarkers of CDK4/6 inhibitor sensitivity or resistance. Thus, there is critical need to identify biomarkers to determine which patients are most likely to benefit from CDK4/6 inhibitor treatment. Here at Olaris, we investigate metabolites as potential biomarkers, and metabolomics is at the heart of our technology. Metabolomics is a study of small molecule metabolites that provide energy and biomass for life to exist. Unlike other biomarkers that relay information about what could be happening or what is involved in the process, metabolites are essentially an actionable readout for what is happening in a patient. At Olaris, we use NMR and MS-based metabolomics and machine learning to identify metabolic signatures that are altered in response to drug treatment or resistance. In this study, we aim to identify metabolic changes associated with the response or resistance to CDK4/6 inhibitors such as Palbociclib and Abemaciclib. Using drug-sensitive and resistant human breast cancer cell lines, we sought to achieve two main objectives. First, to identify metabolic changes associated with PALBO and ABEMA treatment, and second, to identify the metabolic changes associated with resistance to either drug. Resistant cell lines were derived by exposing parental MCF7 cells to progressively increased doses of PALBO to create a PALBO resistance or PDR cells or ABEMA to create ABEMA resistance or ABR cells. Biological triplicates were prepared for each condition, wherein parental and resistant cell lines were incubated for 24 hours in DMEM media supplemented with C13 level glucose and treated with either DMSO, PALBO, or ABEMA. Resistant cell lines had a drug washout prior to the treatment. After incubation, the cells were counted and harvested, and intracellular metabolites were extracted via methanol and chloroform. Metabolites were analyzed using 2D HSQC NMR spectroscopy, which provide a resonance for all carbon molecules attached to a unique proton where the intensity levels are correlated with concentration. MCF7 cells treated with PALBO or ABEMA had significantly different intensity levels of metabolite resonances compared to DMSO treated cells, and they also had different levels of metabolite resonances compared to one another. Resonances were annotated by comparing their chemical shifts to a library of known metabolites, which is highlighted in the tables. The resonances that were not matched to labeled as unknowns and tracked by their proton and carbon positions. One unknown resonance was significantly increased upon both PALBO and ABEMA treatment. In addition, CDK4/6 inhibitor treatment caused a marked decrease in nucleoside and nucleotide metabolites such as adenosine, uridine, and AMP, which is again consistent with ability of CDK4/6 inhibitors to prevent cell cycle progression. Notably, glucose 1-phosphate or GLU1B was increased following ABEMA treatment, but not following PALBO treatment. PALBO and ABEMA resistant cells also had significantly different metabolite resonances compared to DMSO-treated MCF7 cells, and they also had different levels of metabolite resonances compared to one another. After annotation, we found three common metabolites altered in PDR and ABR cells. Glycerol was decreased in both resistant cell lines, but we observed that dihydroxyascorbic acid was increased in PDR and decreased in ABR cells. Serine was the opposite, decreased in PDR and increased in ABR cells. In PDR and ABR cells, we also observed that both ATP and AMP were decreased and glucose 1-phosphate was increased. Pairwise analysis suggested specific metabolite signatures for each drug-treated or resistant cell lines. This was further supported by PCA, where MCF7 cells treated with DMSO clustered separately from PALBO-treated and ABEMA-treated cells. And additionally, PDR and ABR cells also clustered separately from each other and from parental cells. Of note, PDR and ABR cells treated with CDK4/6 inhibitors were significantly overlapped, suggesting minimal changes, which is consistent with the drug resistance. We then sought to compare metabolite levels across all conditions. We found that CDK4/6 inhibitor treated and resistant cell lines had lower levels of ATP and AMP compared to parental MCF7 cells treated with DMSO. In contrast, GDP was significantly increased in drug treatment and resistance. GTP to GDP ratio is a known readout for cell proliferation, where normal dividing cells have ratio near 10. And indeed, in parental cells, the ratio was about 8. This ratio significantly dropped to 1.1 and 0.3 in PDR and ABR cells, respectively, which is then consistent with the decreased proliferation rate of the resistant cells. Glucose 1-phosphate was significantly increased in both PDR and ABR cells compared to parental MCF7 cells, while in contrast, serine was significantly increased in ABR cells but significantly decreased in PDR cells. This could suggest that there are shared and distinct pathways of resistance. Glucose 1-phosphate was increased in both resistant cell lines. Glucose 1-phosphate is an important metabolite in galactose metabolism, or also known as a Lillard pathway. It would be interesting to determine if this pathway plays a significant role in CDK4/6 resistance. The unique increase in serine in ABR cells could also suggest specific upregulation of serine glycine pathway in ABEMA resistance. We are currently assessing whether inhibition of upregulated pathways in PDR and ABR cells can restore CDK4/6 inhibitor sensitivity, which could lead to novel treatment opportunities. Further, we are evaluating whether the altered metabolites in the resistant cell line correlate with metabolites from clinical samples from patients non-responsive to CDK4/6 inhibitor treatment. In summary, our results indicate that glucose metabolism is altered following PALBO and ABEMA treatment, and that resistance to each drug may activate different metabolic pathways. To the best of our knowledge, this is the first direct evidence of PALBO-specific and ABEMA-specific metabolic signatures. And with this, I look forward to engaging in more discussion, and I'm happy to answer any questions or discuss any collaboration opportunities. Thank you.